3, 2, 1. Hello and welcome, everyone. This is the CyVerse webinar series, and I'm Tina Lee, CyVerse's User Engagement Officer. This is our final webinar of the fall and features Zidang and Shivani Chiranjeevi on their work on two apps for the AI Institute for Resilient Agriculture, or AIRA, which is a collaboration between Iowa State University, the University of Arizona, and other institutions. Z and Shivani will be presenting their iNaturalist Scalable download app and a mobile phone app that uses Scalable download to answer the eternal question of farmers and picnickers everywhere around the world: what the heck is that bug? So for information on our webinars and to view past webinars, please be sure to bookmark our webinars page at www.CyVerse.org/webinars. As CyVerse moves towards a diversified funding model, we are supported in part by subscriptions to our platform. These free webinars remain an important component of our mission to deploy a national computational research infrastructure for scientific data-driven discovery. Let's first take care of some housekeeping and then on to Z and Shivani's presentation. Our webinar today is about 30 to 40 minutes long with time for your questions at the end. Please open the Zoom chat window and type your questions there, and Z and Shivani will answer those after their presentation. All webinars are recorded, and I'll post this video recording on our website shortly. Today, CyVerse now has nearly 70 webinars organized into playlists on science, technology, and CyVerse platform topics to help you learn and do your science. You can find those on our website webinars page. And now I'm pleased to introduce our two presenters, who are collaborators in the AI Institute for Resilient Agriculture. Zidang is a doctoral student in the University of Arizona's Electrical and Computer Engineering Department and a researcher at the U of A's Data Science Institute. He received both his BS and MS in electrical engineering at UC San Diego, with a focus on signal and image processing. Shivani Chiranjeevi is also a doctoral student participating in AIRA. She's based at Ames, Iowa at Iowa State University and is in their mechanical engineering department. Shivani specializes in artificial intelligence, image processing, and computer vision in plant sciences. I am so pleased to welcome you, Z and Shivani.
Hello, everyone. Thank you again for coming today. Today, we're here to present to you about how to incorporate iNaturalist species images in your research, machine learning research, and really anything you're interested in.
Hi. Our project is funded by IRA, which is AI Institute of Resilient Agriculture, like Tina said, and Dr. Bhaskar Ganapathy Subramaniam, who's also my advisor, is the director of this institute here at ISU. And IRA will transform agriculture by creating a new AI-driven framework for modeling plants, crops at various agronomically relevant scales. We accomplish this by introducing AI-driven digital twins that fuse diverse data with domain knowledge of experts. A digital twin is a virtual representation that continuously assimilates sensor updates and also respects contextual knowledge to mirror the life cycle of its corresponding biological system. Through a strong social science focus will ensure adoption of AI in agriculture. We also build on NSF and USDA NIFA investments to democratize access to AI tools to broader research and ag community. We also have many signature education activities that will build on AI-aware workforce and to provide AI training to Native American communities and ensure increased participation of women in ag. Finally, IRA will serve as a nexus for broad adoption of AI in ag through activities with our large stakeholder network. So, what are we going to present today? So we have built a mobile web app that can classify 142 agriculturally relevant insect species. This functionality would soon be extended to 2,500 species. So now let's look at some cool features of this app. The first one being its ability to differentiate between beneficial and harmful insect pests. Also, in many insects, they go through metamorphosis. That is, it passes through a series of phases like nymph, larva, pupa, etc. The insect app can recognize these insects at various stages. The next case is where there are insect pest classes that look very similar to each other. These are called fine-grained classes. The app can differentiate between these fine-grained species as well. Finally, though farmers and agronomists deal with insects almost every day, it's often very useful to have common names when you're out in the field for ease of use. So how is the data set organized for our problem? So to understand how it's done, we need to go over a few terms, the first one being the taxonomy, which is a branch of biology that focuses on classifying and naming organisms based on the evolutionary relationships between them. And there are 7 levels in this phylogenetic tree that you can see on the right-hand side, starting from domain, kingdom, and all the way down to species. So our classification is at the species level. So it requires images to be present at this last level in the phylogenetic tree. Normally, the folders are nested in this hierarchy up to the species level. We see one example of how the species Japanese beetle is organized in the taxonomic hierarchy. So what does it mean to to download all the images by myself from a public repository like iNaturalist? So one should have extensive knowledge about the metadata of the images, the structure of the data, which is ordered in the taxonomic hierarchy, compressing the folders, and finding efficient methods for storage and transferring easily. So also we should figure out a way to parallelize these downloads, else the downloads would be very slow. From the iNat website, downloading the data set by image querying is very time consuming, and it's almost very impractical if I would have to download millions of images for a problem like mine.
So we at the Data Science Institute came up with a potential solution to this problem to help the IRA project. And it is something we call iNaturalist Scalable Download. So iNat SD is how I shorten it, is a workflow pipeline tool we created to aid people in downloading subsets of images from iNaturalist that they desire for research purposes with a particular focus on ML research. It's primarily built using the software seen in the bubble here. And so what does it do? The purpose of the tool is to allow users to download all observations of species and subspecies that are under a specific taxon's taxonomic hierarchy. Users will input the scientific name of a specific taxon to the tool. Then the tool will run a Python pipeline to download the species observations associated with that taxon. This allows users to gather vast quantities of high-quality labeled data of organisms from iNaturalist for research purposes. So a little more on iNaturalist. iNaturalist is a community science or citizen science platform where people who engage with the platform record observations representing encounters with individual organisms. So as a result, iNaturalist contains one of the largest stores of labeled images of living organisms in the entire world that's available to the public. It contains over 115 million images and has 2.5 million registered users that are contributing to this repository. Shown to the right is a quick look at what the platform's website looked like. Prior to 2021, it had been difficult to utilize iNaturalist for research purposes due to licensing issues and them limiting download quantities on their API due to infrastructure costs. However, these problems were solved in 2021, where iNaturalist collaborated with Amazon as part of the Amazon Open Data Sponsorship Program to release the iNaturalist Open Dataset. This dataset contains all licensed observations of organisms from iNaturalist, namely, specifically the ones that follow the Creative Commons license, which means that all photos in the dataset can be used for academic research purposes. Another benefit of this sponsorship was that Amazon now covers the cost of the storage and bandwidth fees associated with the dataset. With this dataset, iNaturally also specifically made a push for users to license their images, and now over 70% of the images on their platform are under the Creative Commons license, meaning that this dataset contains over 70 million images. The dataset can be accessed through 4 metadata CSV files that contain the meta information about every single observation in the dataset. The column of each CSV file can be seen to the left, and in bold are the columns that we specifically use when creating iNat SD. For the format and how to access these images, each individual image can be accessed through a URL in the following format. And the URL directly links to the AWS site where the image is shown and can be downloaded. There are 6 different sizes of the images, the biggest being the original image, and then each other is a scaled down version of the image and a unique square image, which is exactly 75 by 75. Our tool, iNat Scalable Download, is an extension onto the iNaturalist Open Dataset that allows users to easily download subsets of the dataset based on a particular taxon group that they're interested in. The aim of the tool is to allow users to easily download image datasets from iNaturalist without needing an extensive knowledge about the iNaturalist data format, SQL, or even programming in general. We also aim to make the data acquisition easily reproducible and documentable, and include various features specifically aimed towards data analysis and ML research. So here we're going to present a demo on how to use the iNaturalist Scalable download. So say that you've now downloaded the repository of iNaturalist Scalable Download. So we see that's the folder the repository is. And say we want to download silver-lined butterfly images. So just all images in iNaturalist of silver-lined butterflies. So all of these things. We now know that the scientific name is Sigaritas. So we use the command snakemake, and then we type the name of the CSV file that we want to create. And this actually triggers the Python pipeline to download all species CSVs associated with Sigaritas. The -- cores argument indicates that we want to use all available cores on our machine. And then we use the -n argument to first indicate that we want to do a dry run, but then we execute it and we see what we have to do is we type in the scientific name of the taxon that we're interested in Sigaritas. So now this is executing. It takes a while to make the data set and download the CSVs. We're going to skip ahead in the video. So now that job has now been completed. And we can now see that the database has been created of silver-lined butterflies, and CSVs for each species under the silver-lined butterfly taxonomy has been created as well. Next, we will use another snakemake command to trigger the Python download pipeline that actually downloads the images. And we do this by specifying the name of the resultant YAML file containing the information about the download. Once again, we use the -- cores argument and do a quick dry run to make sure everything looks good. And then we'll start the download. So now the download is in process. We can actually see that the download is downloading in parallel using all of the cores available on my machine. And we can see that it's providing information about each species and each download. So this also takes a while. So we'll once again skip ahead. So now all of the downloads have finished for specifically silver-lined butterflies, and they're all resultant in this image folder file. So we can go to a specific species, and now we can see that we have all the images associated with that particular species that are available within iNaturalist now. So The next thing I want to talk about is the Insecta dataset that we at the Data Science Institute downloaded and acquired for the mobile insect identification app. Probably the biggest thing about this dataset is that it is incredibly large and contains a vast amount of data. The dataset totals over 14 million images across 95,000 species and is a total of 57 terabytes. So here are some just brief examples of the insects in the species. And with how large the data set is, it's not really possible to store it on local machines. So we use CyVerse and CyVerse's web storage abilities to store the data set, and it also allows users to easily access the data set for their model usage. So while iNaturalist is iNat SD is fully capable of being run on your local machine, like we saw in the example video with the Silverline butterfly downloads, it's also very easy to scale for when you have access to more amounts of computational resources. So this in particular is very helpful for downloading extremely large data sets, such as the Insecta dataset. Like shown in the video, you can specify exactly how many cores in your job execution. iNat SD is designed in a way that considers each separate species download as a separate job so that it can fully utilize the inherent parallelization capabilities within SnakeMake, which is the workflow management tool that iNat SD is based off of. The bottom left shows us the dry run of the Silverline butterfly download. We specified the number of cores we wanted to use in the overall execution. And then we can see that in this dry run, the tool queues up over 150 jobs. Each job is then assigned to one of the available cores for parallelization. So this parallelization can speed up the download times by many factors. So additionally, due to the fact that AWS completely covers the bandwidth costs of downloading, since it's part of the iNaturalist, the Open Data sponsorship, users can actually launch as many nodes for downloading and parallelization as they want, with their only limit being their own computational resources. So for example, in our downloading of the insect identification set, we use the University of Arizona's high performance computing resource to download the images. We created 5 separate instances of HPC virtual machines, each with 20 cores. And to download the entirety of the 57 gigs, it took roughly 50 hours to complete on average for throughout the job executions. So to go into a little bit more detail about that, this slide quickly gives the run through of the overall data flow process for downloading extremely large data sets utilizing CyVerse and your university's HPC. So like the example shown in the example video, first, the user must input a taxon query, and this taxon query will download all the CSV files associated with that query. What then can happen is you upload these CSV files to CyVerse, and then using the very friendly to use iRods command, you both allows for the uploading as well as the download from CyVerse onto your actual HPC. So once it is on HPC, you can basically then use the HPC's computational resources to loop each species download, and then upload, once you're finished with each individual download, upload the files, image files back onto CyVerse as well. And so it basically utilizes the HPC's computational power, but then stores it into CyVerse as utilizing CyVerse's data store power. So you don't clog up either your local machine or your school's high performance computation center. Some additional features that we're currently implementing in iNat SD are shown here. And these are features that are provided in addition to the query download pipeline. First, we have data visualization features, such as the one on the left, which is a sunburst visualization of the top 100 species in the iNaturalist Open Dataset. How a sunburst works is the parent hierarchies start from the center of the circle, and the size of each wedge of the ring is determined by the ratio that group has compared to its parent level. We can use this visualization to determine which species and their associated ancestries in iNaturalist open data have the most amount of observations and the most representation so that we can make decisions in building our model. Another feature that we've implemented is the update feature. So as seen on the bottom right image, the iNaturalist dataset is constantly updating with new images. As a researcher, we don't want to download the entire dataset every time new images are added to iNaturalist. So we created the update feature so that it can compare and contrast the existing images you own of your taxon query with the updated iNaturalist dataset and only download the images you do not have. The next part of the webinar, we will be going over in detail about a successful application of the iNat SD tool, namely its benefits for the mobile insect identification app developed by IRA.
Okay, so why are we concerned about insect identification anyway? So the state where I come from, Iowa, it is very agriculturally rich, and the insect pests cause infestation. They destroy hectares of crops annually here. So this leads to crop damage, food scarcity, and also economic losses to farmers. So we need to know what we're dealing with before moving on to mitigation strategies, because sometimes some insects could actually be beneficial to the crop and you don't want to end up spraying pesticides all over it. So extremely accurate classification is very important. And there are billions of insects on this planet, but we identified 2,500 agriculturally important insects, which is still a very large number, and we developed a classifier to identify them. Most of the state-of-the-art classifiers require huge amounts of labels to train a highly efficient classifier, but labeling is extremely time-consuming, laborious, and oftentimes very expensive. So we propose using label-less pre-training, which is often known as self-supervised learning, which is a branch of deep learning that does not use labels. We use this for a pretext task like learning useful representations that differentiate different classes. And these representations learned during the pre-training is then used for downstream tasks like classification in our case. So now we have to address the challenges that come in the way of developing a classifier. The first one being the huge number of classes. We're not dealing with 10 or 100, but essentially 2,500 different classes. And with increasing number of classes, getting a classifier with high accuracy becomes increasingly difficult. The second one being the fine-grained classes. So there are many classes which look very similar to human eyes. So in the second figure, you can see 4 different species of ladybug beetles. They're completely different, but they look very similar to the human eyes. And contrary to the popular belief that all ladybugs are beneficial, the beetles with lesser number of spots are usually beneficial, while species like 22 spotted ladybugs are harmful. And in the third figure, you can see 3 different images which look completely different, but they're in fact images of the same insect, tobacco hornworm, but taken at different life stages. So the app should be able to identify all of them as belonging to the same class. And during our testing and inference, we were able to verify that our app works well for all these cases with a high accuracy. So story time. So here at ISU, we have an agronomist who is my advisor, Dr. Aarti Singh's colleague. He got the QR code for our app a couple of months back, and one day he found this insect that you see on the slide on his garden, and he thought it was harmful and he wanted to kill it immediately. But he thought maybe I should try this app before I kill the insect. And he found out that it was goldenrod soldier beetle, which is actually a beneficial insect and not harmful because it eats up other harmful bugs. So this is one case where even agronomists who work with plants and insects every day often get confused with such species because you have many insects which are very similar looking in nature, making it almost impossible for humans to differentiate and remember about every insect that's out there. So we saw what the challenges are if we would have to download all the images from iNaturalist website by ourselves. So how does these iNat OpenSD help us with these challenges? With iNat SD, we are now able to download images in a single folder at any taxonomic group that we want. Example, say we want all images of Japanese beetle, which is at species level. We query for this species and get all the images corresponding to that species. Say if there are many subspecies within that species, it will group them together as well. So if a user is interested in genus level organization for his or her problem, he or she could simply query using the genus name, and it would group all species within the genus and give us the images within that genus. For our application, we are interested in species level classification, and this folder organization that you see on the right-hand side, where we have all folders named after the species, and we have images of the species within that folder. This is exactly how the dataset module of our classifier expects the dataset to be organized in. So it's very easy to integrate with ML and deep learning applications. Also, the users have an option of downloading the images in any resolution they desire according to their needs and the computational resources they have. So the dataset details for the classification, the dataset size we're using is 2 million images. The number of classes we are targeting in the image insect app that I'm going to show you is 142, but we also have a model in place that classifies 2,500 insect species, and it would eventually be deployed soon. The input resolution we chose is 224 cross 224, and the reason why we chose this was because usually higher resolution images demand more computational resources, whereas lower resolution images lower the accuracy of the classifier. So keeping this compute power versus accuracy trade-off in mind, and also the fact that many popular CNN architectures like ResNet 50 adopt the same resolution, we choose 224 cross 224. So for our classifier training, there are two stages. The first one is the self-supervised pre-training, followed by the classifier fine-tuning. So the first phase is where we use label-less self-supervised pre-training. And here we use 2 million images for an SSL method called SWAV, which is swapping assignments between multiple views of the same image, which is an online contrastive-based clustering method. We ran this model for 6 days on 4 nodes of 800 GPUs, each having 4 GPUs, totaling to 16 GPUs. This model, the model which classifies 2526 classes, which would eventually be available in the future, uses more images, close to 6 million. And that model, with the same amount of resources, took us close to a month for pre-training, which is a long time. And coming to the next phase, which is the classifier fine-tuning. For this, we used one 800 GPU, and it takes close to 3.5 days for 2 million images. This classifier has 142 classes. Coming to the performance of the classifier, we have a top 1 accuracy of 97%, a top 5 accuracy of 99%, and a mean per class accuracy of approximately 96%. And on the right-hand side, you can see the histogram of per class accuracies plotted against the number of classes. And you can see that most of the classes have per class accuracies lying between 95% to 100%. We now compare our classifier's performance with the classifier developed by iNaturalist community. So they reported a top 1 accuracy of 77.1% for the Insecta class, which has 2526 classes. And when we compare it to our classifier, which gave us top 1 accuracy of 92.6%, you can notice that there's a significant improvement in the accuracy, close to 20% improvement. Now I'm going to show you a demo of the Insect app to see how it looks like and how it works with the help of an example. So first you click the choose file button and you upload the image of the insect. It would display the common and scientific names. First one, we have a beneficial insect, which is the goldenrod soldier beetle. And for the second example, we have Japanese beetle, which is harmful to soybean, and we see this a lot in Iowa. So an insect that I've become fond of recently. It's not good for soybean, but it's very shiny and pretty to look at. And the next one, Ozzy.
Yeah, we will talk about some of the future work that we have planned, both for iNat SD, the iNat download tool, as well as for the mobile insect identification app. For iNat SD, something that we aim to be able to do is to include more dedicated pre-processing options in the download itself, and more flags for people to choose from so that it can be easily plugged in to popular architectures. Another aspect we are thinking about is to integrate the location data of the images through heat map visualizations for each observation and for the data sets they choose, so they can use that information to determine, make decisions about their model. And then something we're interested in and exploring is investigating the possibility of automatic bounding box labeling for the output data sets, and basically trying to explore what the bleeding edge is in regarding that and how it can be applied to what we have with iNat SD.
And like I said, the app that I showed classifies 142 different insect pest species, but we plan on releasing an insect identification mobile app for 2,500 classes early next year. And also we are collaborating with entomologists here at ISU to incorporate integrated pest management strategies for each species. So say my app gives the classification result saying it's a particular insect, say Japanese beetle, it will also display strategies on what you would have to do when you encounter the same. So it's going to be very handy for farmers to walk around the field, capture an image, and it's going to tell them what they would have to do. They wouldn't have to carry very huge manuals around anymore. So
finally, we also want to give a quick shout out to CyVerse and just kind of how amazing it is, both as a actual resource for like data storage, data acquisition, and data management. Like I mentioned, the Insecta Dataset is 57 terabyte. It's very big, and nothing I have could come close to storing everything, all of that. But we were able to do so utilizing their service and able to incorporate it into a practical application thanks to CyVerse effectively. And of course, CyVerse also does wonderful webinars and resources such as this, and teaches people about the great advances in data science, as well as a variety of fields. So shout outs to CyVerse. And that is the end of our presentation. I think if there are any questions, we have emails for people to send their questions to, but also we have time right now to answer any particular questions that people have. And I see that there are already some on the chat, so I'm gonna bring them up now. And so do you see them as well, Shivani?
yes, I think I can answer the first question that from Tina.
Yes.
So the question was, what do you attribute your higher ID accuracy to? So the accuracy would mostly be attributed to the pre-training that we do. We do spend a significant amount of time during for a classifier on the pre-training, and we've noticed, we've compared our models without pre-training, and we noticed that the higher accuracy is because of the pre-training, and also in conjunction with the other state-of-the-art augmentations and a couple of other techniques that we've used. But the main thing would be the self-supervised pre-training.
Great, thank you. Feel free to type in questions if you have them. I have some other questions like, when will this be available? Where will it be available? Just kind of where you are in releasing the app.
Okay, so the app is currently hosted on CyVerse. It's hosted on our local ISU server here, and for 142 classes, and we plan to make this available early next year, say Jan to March, and it should be available. It should be available on CyVerse as well as on ISU's website.
Mhm. Similarly, iNat SD will also be available around that time, and it will also be, it will likely primarily be accessible through the Data Science Institute's GitHub at University of Arizona.
Okay.
But
So
more information we'll talk about closer to release dates and such.
okay. So as I take it, this is the open source code that somebody could fork and replicate to deal with other species that are available on iNat. Open Dataset.
Yeah, and the goal is even without modification, hopefully they'll be able to kind of do some sort of task in that sense that they want as well. And since it's open source, if they have ideas for improvement for it's kind of feel free to do it kind of idea.
Okay. Are there any other questions besides me? Feel free to type them in. If not, basically this is the end of our webinars for this semester. And we thank Z and Shivani for creating this wonderful apps and that take advantage. Oops, somebody has a quick question. Please, Rob, if you'd like to unmute, you're welcome to. Otherwise, feel free to type it in. Oh, okay. What kind of biases do you expect from the iNaturalist dataset versus field applications? Are many citizen scientists photographing, for example, sharpshooter pests? the
answer that. So the biggest thing is that it is an unbalanced data set in a sense that the most kind of popular, interesting insects get more representation because it is a citizen science platform. So the easiest example is that the most popular insect, for example, are bees. There's many bee photos, but for something like sharpshooter pests, it will have less data. But because not only citizens, or in a sense, so people very interested in research also post their observations onto iNaturalist as well. So you end up kind of with a large spectrum of types of images.
Okay. RT says our app can identify leafhoppers, though, for sure.
So yeah. So I think the answer that I have is that while the biases do exist, knowing about the biases, you can adjust kind of the data set that you use and your you can adjust your model to account for it and subset your data in ways to basically address those biases and have your model still perform the best it can. So that information is basically, it's part of the so to answer Bill's question, who vets the images for classification. So that is all on iNaturalist's side. So basically iNaturalist has a very kind of pretty strict system in how they actually identify an image. And they have categories on how likely how trustworthy basically an identification is. And so in the iNaturalist open data set, you can specify what types of what level of identification that you want in your research data. So basically they have something called like research grade observations, which basically an observation identification only counts as research grade if it has multiple verified and kind of iNaturalist qualified users to identifying the data. I will, so the people themselves are the users of the iNaturalist platform. So iNaturalist is quite popular, specifically, especially kind of in the biodiversity and life science field. So generally, it's a system of the more kind of correct and good classifications you do on that platform, the more reputable you become in that in the iNaturalist website platform. Yes, we only apply research grade data in our training. It's one of the flag options in downloading.
That's what makes your accuracy so high too, right? Yeah, okay. If there are any more questions, type them in. Otherwise, really, Z and Shivani, thank you so much. Oh, how appropriate might this technology be for field ID when not connected to the internet?
So field ID as in field identification. I guess, Shivani, do you want to tackle this question?
Yes. Currently, we're planning to quantize the model, that is, make the model very portable and handy on mobile devices, so that, you know, once you download the image, the app, other than times during updates and other things, you should still be able to use the app without pretty good without internet connection. But that's something that is in the pipeline. We currently don't have an app that would work without internet connection, but this is definitely in the pipeline, and we hope to get it done soon.
Okay. Any more questions for Z and Shivani? Great work, you guys. Thank you so very much. And thank you to RT Bhaskar and Nirav for encouraging and guiding Shivani and Z in this great user-friendly app. Oops, if you have an image, you can use it where internet is available, says RT. So, okay. All right. Well, then we will see everyone hopefully back next spring when we have a new slew of webinars for you. Until then, take care, everyone. See you back online. And thank you, Z and Shivani and the ERA Institute people for helping to present this app.
Thank you, everyone.